Welcome to our exploration of DNA structure, the molecule that carries our genetic information. DNA takes the form of a double helix, which can be visualized as a twisted ladder. The sides of this ladder are made of alternating sugar and phosphate molecules, forming what we call the backbone. The rungs of the ladder are made up of pairs of nucleotide bases. These bases follow specific pairing rules. There are 4 types of bases: adenine, which always pairs with thymine, and guanine, which always pairs with cytosine. These base pairs are held together by hydrogen bonds, shown here as red lines. AT pairs have 2 hydrogen bonds, while GC pairs have 3, making them more stable. This remarkable structure was discovered by James Watson and Francis Crick in 1953, revolutionizing our understanding of genetics. When viewed from a different angle, we can see how this ladder-like structure twists to form the characteristic double helix. This basic structure is essential for DNA's role in storing and transmitting genetic information. Each DNA nucleotide consists of 3 essential components. First, we have the phosphate group, which forms part of DNA's backbone. Next is the deoxyribose sugar, which gives DNA its name, deoxyribonucleic acid. Finally, we have the nitrogenous base, which carries the genetic information. DNA uses 4 different types of bases to store genetic information. Adenine, represented by A, always pairs with thymine, represented by T. Guanine, represented by G, always pairs with cytosine, represented by C. The sugar and phosphate groups alternate to form the backbone of the DNA molecule. The nitrogenous bases attach to the sugar molecules, pointing inward in the DNA double helix. During DNA replication, the first step is breaking the hydrogen bonds between base pairs. This process is carried out by an enzyme called helicase, which acts like a molecular zipper. As helicase moves along the DNA molecule, it breaks the hydrogen bonds between base pairs, separating the double helix into 2 single strands. To prevent the separated strands from rejoining, proteins called single-strand binding proteins attach to the exposed single strands. The region where the 2 strands separate is called the replication fork. This creates 2 template strands that will be used to build new DNA molecules. DNA polymerase is the main enzyme responsible for synthesizing new DNA strands. DNA polymerase can only add nucleotides in the 5' to 3' direction. On the lagging strand, DNA synthesis occurs in short segments called Okazaki fragments. DNA ligase then joins these Okazaki fragments together to form a continuous strand. This combination of continuous and discontinuous synthesis allows for efficient DNA replication in both directions simultaneously. DNA polymerase has built-in proofreading abilities to ensure accurate replication of genetic material. As it synthesizes the new DNA strand, it carefully checks each newly added nucleotide against the template strand. When an incorrect nucleotide is added, the polymerase detects the mismatch. The proofreading mechanism involves 3 key steps. Once replication is complete, the process results in 2 identical DNA molecules through semi-conservative replication. Each new DNA molecule contains 1 original strand and 1 newly synthesized strand. This ensures that genetic information is accurately passed down to daughter cells.